My name is Matt Gilbert. I study thermal physiology in the Farrell lab under Tony Farrell at the University of British Columbia. And I'm really excited to be here today to talk about some work I'm doing in the Central Arctic with Les Harris and Brendan Malley from Fisheries and Oceans Canada. They're here at the conference. And with J.S. Moore from University of Laval. So I'll start by just explaining why I've focused on Arctic char. As Les went into a lot of detail on yesterday, they're the focus of a large commercial fishery in the Canadian Arctic. And this photo just shows what a typical harvest looks like in the Kitikmeot region of Nunavut where I work. And they've also had a long history as a subsistence fishery in the Canadian Arctic. And this is actually one of the images that Paul Vesci was talking about last night when he was talking about the field site he studied Arctic char at. And this image was taken in 1916 by the Canadian Arctic Expedition and shows a group of Inuit fishers at a rock weir spearfishing. And it still serves as a subsistence fishery today. This is one of my field assistants having just processed a Arctic char in a traditional manner where the fish is hung to dry. And for those of you who've experienced this, had a chance to work in the north, it's a really incredible experience. And then from a more physiological perspective, why I'm interested in char, they're kind of typically thought of as a very cold adapted classic example of a stenothermal animal that requires cool, well-oxygenated waters throughout its life. And that's really in stark contrast to the trends that we see in global warming, where warming is progressing much more rapidly at high latitudes than at more temperate latitudes. So this figure here just shows the average temperature anomaly over the last 10 years relative to 30 years in the middle of the last century. And you can see that at high latitudes, including our field site, warming has occurred at nearly twice the average global rate and twice the rate of temperate areas where a lot of the research on salmonid thermal physiology has been done. So that was at a global scale. At a more local scale, we're already seeing examples of these fish experiencing really drastic thermal variation and incredibly warm temperatures. So this is a figure from some work that I was fortunate to be a part of during my master's on Nullahugget Creek, again the system that Paul talked about yesterday. And what we see in the first panel is kind of what a typical Arctic stream that bears char would look like at freshet. And then as the summer progresses, this particular stream discharge decreases rapidly during the course of the migration to the point that it's completely inaccessible by the end of the summer. And that's shown by this discharge plot here. And what I really want to highlight is because of the shallow nature of these streams, the fact that there is no coverage and there's a relatively large surface area exposed to ambient temperatures, they experience really drastic fluctuations in temperature. For example, here we can see a fluctuation where in a single day it goes from around 10 degrees, 11 degrees in the morning, up to 21 degrees by the afternoon. And so that's just concerning on its face, but it's even more concerning when you consider what we know about the physiology of anadromous salmonids. And thankfully that's lowercase. I had a minor panic attack when I saw that. So this particular figure, or something like it, has been presented a couple times throughout the conference. And what we're looking at here is the ability of the animal to uptake and use oxygen as temperature increases. And the dashed line here is the resting oxygen consumption. And we can see that it increases exponentially as temperatures rise. And on the top we have our maximum oxygen consumption that the animal can sustain. And it increases to a point at which it plateaus and begins to decline. And the difference between the resting and the maximum is the scope or amount of oxygen available to the animal to perform functions that are not essential, such as migration or any form of activity. And the reason that we think that this maximum oxygen consumption and aerobic scope becomes limited at high temperatures is because maximum heart rate also becomes limited. So in this plot here, we're looking at maximum heart rate over temperature of coho and Atlantic salmon, just as an example. And what we see is that it increases to a peak or plateau, and then there's a period of cardiac collapse after which the heart actually becomes arrhythmic and starts skipping beats, and that's indicative of cardiac failure and would eventually be lethal if it persisted for long enough. So given these concerns, the central question that's really guiding my work in the Arctic right now is do thermal limitations to aerobic and cardiac performance have the potential to reduce migration success in anadromous Arctic char? And within that question, I've come up with a few study objectives for the particular work I'll be talking about today. The first is just to provide a field-based assessment of the thermal limits of cardiorespiratory performance in migratory Arctic char, and then compare those limits that I identify to the actual environmental conditions that these fish are encountering to identify potential thermal barriers to migration from a physiological perspective. And then the third is less related to the biology of the fish itself, but traditionally these methods have been confined to areas where you have access to a larger research institute and a lab. So we're validating and demonstrating the utility of some mobile research infrastructure to allow us to conduct this research in very remote settings. So my study area for this particular site is the Kitikmeot region of Nunavut that Les Harris highlighted in his talk yesterday. And the data that I'll be presenting first is all collected along Freshwater Creek, which is the nearest char-bearing stream to Cambridge Bay. And Freshwater Creek shares a lot of characteristics with the creek I described earlier. There's not a lot of coverage, it's got a lot of air exposure, has the potential to experience large diurnal temperature fluctuations and high temperatures. And the mobile labs that we use were developed by the Arctic Research Foundation. They're a nonprofit organization that's working to develop Arctic research infrastructure. And these labs are converted shipping containers. They're fitted with 15 solar panels each, 2 wind turbines, and a backup diesel generator, and a large battery bank to store power. And this is what the one that I use looks like on the inside. They all have different uses, but this one is specifically made for fisheries biology. And you can see a large 450 liter holding system. It is temperature controlled, and then there's a bunch of other temperature control units to allow me to control temperature of various experimental baths. There's plenty of power to power all of that equipment. And as you can see here, the far wall actually folds right down to move equipment in and out and fish in and out. So for this study, luckily fish capture was done through angling, which, if we're being honest, is why a lot of us got into Charr research in the first place. And then fish were transferred by ATV in a aerated cooler back up to the lab where the experiments were done. So for the actual assessment of heart rate, fish are anesthetized and placed in an anesthetic bath with water flowing over their gills, and we fit them with ECG leads to record the electrical activity of their hearts. And once we have a stable heartbeat and a good signal, we inject them with two drugs, one that's a mimic of adrenaline to basically act as the gas pedal, and the other that blocks the abilities of the fish's brain to slow heart rate, so basically taking away the brake of the heart. And that's essentially the same thing that happens during maximal exercise, so we consider it a pharmacological exercise challenge. And once we have a stable maximum heart rate, we either acutely warm or cool the fish at a rate of 5 degrees per hour, monitoring their heart rate continuously over that time. And I showed you the data for warming in the background material, but this is actually the first time that this study's been done with cooling, and we don't really know a lot about cardiac performance with acute cooling. And many of you will be familiar with what a typical ECG looks like, but a fish ECG, for those of you who don't know, looks almost identical to a human ECG, which is pretty cool, and it lets us use a lot of the same techniques that you can use in medicine. So a brief anatomy review of a fish heart. We have our atrium and our ventricle. The atrium contracts first after receiving blood back from the body, passes it to the ventricle, and the ventricle is the large chamber responsible for distributing blood throughout the body. And in that process, there's an electrical signal of each chamber, and we can see those signals in our ECG. The first being the P wave, which is the atrium, followed closely by the QRS complex, which is indicative of ventricular contraction. As we warm the fish, however, heart rate increases, so those QRS complexes will get closer together, but then we can also start to see arrhythmia develop, where we have an irregular heartbeat, and sometimes we'll have an atrial contraction, but a missing ventricle contraction, indicating that the ventricle is starting to fail. So to get into what the data actually looks like, here I'm showing individual traces to give you an idea of what individual variability looks like and just the range of temperature tolerance that can exist within a population. So from around 5 degrees up to 16 degrees, you can see that there's very low individual variability between our recordings, and heart rate generally increases at a similar rate among fish. Above that temperature, we start to see individuals plateau and decrease, and that occurs at different temperatures for different individuals. With cooling, what was really exciting, this is the first time we did this, as I mentioned, we did see a similar trend at cold temperatures where heart rate, the rate at which heart rate was changing, began to change at or become more variable at cold temperatures, and heart rate actually started to plummet in individuals as we reached near freezing temperatures. So to look at this, to linearize this, we look at the lawn transform data and the average data. And I'll start by going over the warming data here. In the phase where heart rate is increasing fairly consistently, we can actually analyze this for breakpoints that indicate a change in the rate at which heart rate is increasing with temperature. And that's called the warm Arrhenius breakpoint temperature. And above that temperature, basically, heart rate increases at a slightly lower rate than it was previously. And what we think is occurring there is that heart rate isn't keeping up to the increase in metabolic demand, and the fish starts to get behind there. And because of that, often that temperature will correspond with the optimal temperature for aerobic scope. As we continue warming, we start to see fish reach their peak. And so here I just have the cumulative frequency distribution of the occurrence of each of these events. So we start to see fish peak at around 16 degrees, with most peaking around 18 to 19 degrees. As we continue warming another 2 degrees, fish start to become arrhythmic. And then just for comparison with other literature and more traditional methods of assessment of thermal tolerance, we've included CT max, which is kind of a more crude method where fish are simply warmed until they roll over, and the temperature where they lose equilibrium is called their CT max. And that's another couple degrees higher. So we can see that these heart rate assessments are kind of a more sensitive sublethal indicator of temperature tolerance. And then on the cooling side, again, this is a really exciting finding for me, is that there's also a really clear break point between 3 and 4 degrees where heart rate falls off much faster than we would have predicted. And to really hammer this point home, we can look at the thermal dependence of maximum heart rate using the Q10 temperature coefficient. And for those of you who don't study rates and temperature interactions, all this is looking at the relationship of a change in rate over a change of 10 degrees Celsius. And if we look at the incremental Q10, that means we're calculating it over a smaller increment and then spreading that out to what it would be if it had occurred over 10 degrees. So to put that in more simpler terms, if we have a Q10 of, say, 2, shown by this dashed line here, that would mean a doubling in a given rate over 10 degrees. A Q10 of 1 would mean that heart rate was, or that rate was constant over those 10 degrees. There was no change. And we can see from 4 degrees here up to around 16 degrees here, that heart rate increases at a reasonable rate, but then approaches a Q10 of 1, meaning that it's, on average, it has plateaued, and below 4 degrees, it really shoots up, kind of marking that plummet in heart rate that we see. And because we had those mobile labs, we were actually able to measure aerobic scope in the field. We don't have it for as many individuals as we'd like, but I think we can still pull a couple interesting findings out of this data. So to generate this aerobic scope data, fish are chased to exhaustion in that holding system that I showed before. And once they're fatigued, they're no longer responding to hand chasing. Then they're rapidly transferred into a respirometer where we record their oxygen uptake rate. And then we leave them at their given temperature for 24 hours to recover, and we can get a routine oxygen uptake rate, which is shown in blue. And then again, the difference between the two is their aerobic scope. And so here we were kind of validating that we see the expected changes in aerobic scope the changes in aerobic scope that we would expect based on our heart rate changes. And so basically that is that aerobic performance is maintained over this range. So here we are looking at aerobic scope, and it's relatively consistent over that range. And then above 16 or 17 degrees, where we saw heart rate start to plateau, individuals actually weren't able to recover. So this individual and this individual, denoted by the X's, actually lost equilibrium following the chase and never regained it. So basically, just a 3-minute chase by hand was lethal at those warmer temperatures. And the one individual that was able to recover had a much significantly reduced aerobic scope. So to interpret this in context of what these animals actually experience in the wild, I've overlaid a lot of those metrics over a temperature frequency distribution. And this was recorded from Nullahugyat Creek. It's probably the most extreme example of a warm, or of a system that experiences warm temperatures in our region. And so we see that the optimal temperature, indicated by that first breakpoint, does correspond with commonly encountered temperatures, which is what you would expect. But fish are already experiencing temperatures in some systems that, unless they're able to modify their physiology and acclimate to those conditions, would result in cardiac limitation and even loss of equilibrium in a couple cases. So what's the significance of that? Well, for a fish undertaking an already physically demanding, aerobically demanding migration through these shallow waters, adding that thermal stress could be kind of the straw that breaks their back and could reduce migration success. Avoidance is definitely an option, and Paul talked about some of the unique behaviors that those fish express, not wanting to migrate at warm temperatures. So best case scenario is a migration delay if they encounter those. Worst case scenario would obviously be mortality and complete migration failure. And from the lab perspective, they performed really well. We're really excited about the quality of the data. It was really close to what we can get in an actual lab setting. And the biggest thing that these labs provide is a really stable power supply for any type of research that you'd want to do in the field. So I'll talk a little bit now just about a couple other projects that we have on the go. So I talked about the diurnal variation that these fish experience, but they also experience really drastic variation in thermal habitat over the course of their lives. So we're all familiar with the life cycle of anadromous Arctic char. And so they spend the early parts of their lives in a lake rearing environment. That tends to be a cooler, more stable environment. Mike Power did a great job, or had a great presentation yesterday showing that the overwintering temperatures of those fish in those lake environments can be between 0 and 2.5 degrees, so very cool and stable. When they do migrate out to sea, Les Harris's presentation showed yesterday that their body temperature on average is between 5 and 6 degrees. Again, cool and stable. And then they have these periods where they migrate upriver, and it's a brief period in their life, but it's a critical life history event where they experience those really warm temperatures. So if we're looking at the distribution of those temperatures that the fish might experience, the blue data is lake and ocean body temperatures from data that Les Harris and others have collected using acoustic telemetry, and the red is just river temperatures taken from data loggers placed in streams char-bearing streams in the river during migration. We can see that there's quite a clear division in the thermal regimes that they would experience in those two stages of their lives. So one of the questions I'm really interested in right now is, what is the phenotypic plasticity of these aerobic and cardiac traits between these environments? Are they able to acclimatize to have their physiology suit their environment better? And we know this is the case in other salmonids. And so this is some data that I collected earlier this year in California at UC Davis. And here we're looking at the same maximum heart rate relationships, except in rainbow trout. And we're looking at fish acclimated to 12, 18, and 24 degrees. What we see is a resetting of heart rate, but then also shown over here by the solid blue line is that the temperature at which the heart rate becomes arrhythmic increases by over 5 degrees with acclimation. So Arctic char likely possess some ability to do this. How they actually use it in the wild is something I'm really interested in. And then another project that's ongoing is we're looking at how these traits are locally adapted to the migration harshness that a given population experiences. So here in the West, we have two longer systems that we would consider more harsh that fish migrate through. And then in these two eastern populations, we have fish that undertake migrations of only a couple kilometers. And Les and JS and others have done work that shows there's genetic differences associated with this. So I'm really interested in seeing how these physiological phenotypes play out between those environments. And preliminary data on this, we just have heart mass here relative to body size. And I'll just draw your attention to this figure here, looking at the relative ventricular mass, which is just the ventricle mass divided by body mass expressed as a percentage. And we can see that as we'd expect, and as we see in sockeye salmon that undertake different durations of migration, that fish from longer systems tend to have larger body sizes, or larger heart sizes relative to their body size. And this is just preliminary results. We'll collect the same data on more systems. So with that, I'd like to again just acknowledge the co-authors on this talk and give a huge thanks to our community partners. This work really wouldn't be possible without community support, given how logistically challenging it is and how valuable these fish are to the people in the north. And acknowledge the Arctic Research Foundation and then funding agencies. I'd also like to give a really kind of more thorough acknowledgement to the people who not only keep us safe, but really help with a lot of the data collection in the field. So my field assistants last summer, Colin, shown here, and Darcy. And I'll leave you guys with a little entertainment while I take any questions. Thanks.
Matt, the last figure you presented, could we go back to it for a sec? Just something, yeah, so, okay. I guess my, if I was looking at the figure on the left, I tend to have a reaction that, you know, it's just continuous according to body size. So really, if you have a longer river, you make your body bigger. I see on the right, you kind of show that you have a significant difference. So that's all I wanted to check was I looked at the figure on the left, and then you made the statement. I kind of missed the one on the right. So.
Okay. Yeah.
Yeah.
So we should mention, and I'm sure you're aware, working with the data from up there, that the fish going up Halakvik tend to be the reproductively mature individuals that are going to spawn, whereas there's more fish using the echolux system for overwintering refuge. So I think that's why you may tend to get the smaller individuals using that system, and so they're more represented in that sample.
So since you're at UBC, you probably have heard of some species known as Pacific salmon. And do you have a sense of how this plays out with Pacific salmon? Because they have definitely a wide variety of migratory distances. There's some very nice model systems. A lot of other research has been done.
Yeah, absolutely. So fish, sockeye salmon are kind of the most well-studied example of this. The nearshore populations have smaller hearts relative to body size than the populations that undertake longer migrations. And if you're familiar with the system, Hell's Gate tends to be kind of the division between the physiology of those populations. Populations above Hell's Gate also tend to have higher aerobic scopes, and because of that, potentially higher thermal tolerance. So yeah, it does correspond well to what you would expect. Yeah.
Yes, thank you very much. You mentioned phenotypic plasticity as being of your future studies, so it's very important. But so I just want to ask you what your feeling is about the significance of plasticity's physiological scopes in Arctic char.
Yeah. So based on this data so far, I would think it would be very important. For the fish migrating through the river, the cool breakpoint temperature occurs at 4 degrees, which is a commonly exploited temperature for fish in the ocean and in lakes. So I would think that they would have to be able to shift that temperature substantially lower if they're going to perform well under those conditions. There are other alternatives that they're using other mechanisms for oxygen delivery as opposed to just regulating heart rate. So you can also regulate the volume of blood pumped with each stroke and things like that. But yeah, I would think it would be really important. And there's one other study that was done in Greenland in sea-run char involving John Stephenson, and they found for all of the critical temperatures, they were 5 to 7 degrees lower than what we find here. So there's definitely regional differences, whether or not those are phenotypic plasticity or actually population differences. We'll find out with this work.
I think we'll take one more question from Ben, and then if we have any others, hopefully we'll be able to find Matt at the break or at the barbecue.
Matt, thanks for a great talk. That setup's pretty awesome. I was just wondering, you focused on the freshwater component of this because it's reaching the highest temperatures. If you ran this similarly with salt water, either 15 or 30 parts per thousand, what would you expect to see? And also, how do you think having an anadromous fish, which is coming in from salt water with all the adaptations you need to live in salt water, would compare to one that is in a resident or one that decided not to go out to sea in a particular year?
So there's a lot to unpack there for sure, and we can talk about it at greater length, but to start with, I have done some work at UBC. There's a recirculating aquaculture system that they're looking at to rear fish under different salinities on land-based aquaculture. And so I have done these measurements at 0, 5, 10, and 30 ppt and didn't find any real differences in a lot of the critical temperatures. So salt water alone I don't think would have a large effect. More that the temperature in the marine environment is going to be much lower, so they're going to be thermally acclimated to the lower temperature. And if I can get to your other question one-on-one, I think that'd be good. All right.
All right, thank you.